So we had talked about viruses as a mechanism to deliver genes to an animal cell in biotechnology. And this opens the door to talk about viruses in a more general sense, so we have some information about them. Now, viruses come in many different forms and shapes, but ultimately they're genes in a box. They are a piece of nucleic acid or pieces of nucleic acid, either DNA or RNA, surrounded by a protein coat minimally. Some of them will also have a membrane, but in general, you can think of them as very, very complex chemicals because they're not alive. They don't fit our definition because they're not a living cell. They need to infect a living cell to reproduce their genetic material and then hijack the machinery of that living cell to make proteins and more of their DNA or RNA and assemble more virus. So it's something that no living cell is free from because viruses infect just about everything that's alive. Now, they're very small, right? So here is on the left side a human red blood cell, or just the edge of a human red blood cell. And then there's an E. coli cell drawn in proportion. And then you can see the T4 bacteriophage, which is a virus that infects bacteria. So even prokaryotic cells, even bacteria, can be infected, get sick with a virus, and generally that virus will kill them. Adenovirus, tobacco mosaic virus, plants and animals both can be affected by viruses. The general idea of a virus is to infect a host cell, use that host cell's machinery to make more of its proteins, copy its DNA, and then reassemble and exit the cell. That oftentimes will kill the cell. And what this introduces then is two basic life cycles that we see in viruses. And we're going to use the T4 bacteriophage as our example. And the first of the two life cycles that we see in viruses is called the lytic life cycle. So you can see this is called the lytic cycle of phage T4. And phage, or bacteriophage, is just a fancy word for virus that is specific to prokaryotes. Now, what we can see up here, number one, is the T4 phage binding to the E. coli cell. So there's going to be specificity between the proteins of the virus and the proteins found on the surface of the plasma membrane of the E. coli cell. That's because viruses are oftentimes very species specific, those that they can infect and those that they cannot, and we'll talk about that a little bit more later. The T4 cell binds to the E. coli cell, injects its DNA into the cell along with some proteins. Then those proteins start to chop up the DNA of the host cell, leaving the machinery of the cell then to be used to make more DNA, more viral proteins. All the parts of the virus then are produced, and these will assemble spontaneously. So if you put all these different parts of this kind of little lunar landing module of a virus in a jar and shake it up, you would get these little completely assembled virus particles when you were done. That's just the way it's built. So the virus then can lyse the cell and escape. Now you have many viruses where you had one before. Each then can in turn affect a cell to make more virus. Again, this is called the lytic cycle because it lyses, it's lytic, it bursts open the cell, and it's actively reproducing virus. Now what we're going to do is contrast that then with what's called the lysogenic cycle. And the lysogenic cycle is going to be the key to us using viruses to deliver genes. Because in the lysogenic cycle, this is the cycle on the right on this page, after the virus inserts its DNA into the host cell, as opposed to destroying the host DNA, what it does is it incorporates itself into the host DNA. And this is what we call now a prophage. So remember, a virus that infects bacteria is called a phage. When the DNA of that virus has incorporated itself into the prokaryotic chromosome, we call it a prophage. Literally, it is now part of the bacterial genome. And when that bacteria, which goes on to reproduce itself, making a copy of its genome, we talk about binary fission, right? It's going to make a copy of the prophage right along with it because it is part of its DNA now. Now, occasionally, under the right environmental conditions, that prophage will pop out of the host DNA, and it potentially can go lytic, producing viruses that then kill the cells. But if things are good, if the conditions are right, that prophage will stay in the genome of the host cell, reproducing every time the host cell reproduces. Now, this is the mechanism by which we can use viruses in biotechnology. This is how we do gene therapy. Because if we can take the viral genes out of the virus and put in the genes that we want to insert into the animal cells, in this case our bone marrow cells, into the virus, and then use the virus as a delivery mechanism, that virus will infect the cell that we expose it to and insert its genes, quote unquote, into the host cell's genome. If those genes are the genes that we want to put in there, genes that may correct for genetic disease, we've just genetically modified that host cell. All right, so you can see again now the power of the virus in the mode of being lysogenic, right? Inserting its DNA into the host cell's DNA as a mechanism for gene transfer. So this is very, very useful, very, very important. Now, coming back to this idea of host specificity, as we tie up kind of our things on viruses. So the interaction between the virus and the host cell is very, very important. So this is a more advanced picture of the life cycle of a virus. But what I want you to focus on is this virus in the upper left and the arrow that says attachment. So you can see this happens to be an enveloped virus. So it is DNA on the inside, or RNA on the inside, surrounded by a protein coat. And then they have the envelope around that. That's the circle around that. And the little studs on there are all the proteins. Now, those proteins have to be specific to the cellular receptors. See how they bind? And only when they bind, those receptors bind to the receptors on the virus, can the virus gain access to the cell. So, you may say, well, why are there cellular receptors out there waiting for a virus to bind to them? Well, those cellular receptors do something else. They're important for some other mechanism for the cell to live, but the virus has hijacked that mechanism, so it binds to it and says, haha, I fooled you, take me inside, and then I'm going to take over. And that's how it works. But that is why your dog may have a cold and you won't catch it. Because the virus that is infecting your dog to give it a cold doesn't have the right receptors to bind to your cells. Now, this is the source of what we talk about when we talk about bird flu, because it's a flu that we found in avian species and bird species, that when it mutates and its DNA changes, and hence the receptors change, and all of a sudden where they couldn't bind to human receptors, now they can. Now all of a sudden we have a new virus that can infect humans, and it may be very, very damaging. We don't know, right? So this is how viruses jump from species to species. They have what's called a host specificity, right? And that's that locking key between the viral proteins and the receptors on the cell. Now, what we call is the host range of the virus. Some viruses will have multiple species they can infect, right? So they may be just one in their host range, or they may be many species. But it all comes down to this mechanism. Now, some other things I want you to know about viruses, specifically, are called retroviruses. So retro means reverse. So HIV, for example, is a retrovirus. And that retrovirus, when it attaches to the cell membrane and is brought into the cell, it has RNA molecules. And what it brings with it is a very special protein that we do not have, and it's called reverse transcriptase. So this is one example of a breakdown of the dogma of molecular biology. Because in the dogma of molecular biology, we talk about going from DNA to RNA to protein to trait. Well, reverse transcriptase is a protein that takes RNA back to DNA. It goes RNA to DNA. Reverse transcriptase. Okay, reverse transcription. That DNA that is the product of reverse transcriptase can then be integrated into the host cell genome, and then it can be transcribed and translated by the regular dogma to make more viral particles. Now, I bring this up because if HIV is one of those retroviruses that uses reverse transcriptase, that's a target for antiviral drugs. Right? Now remember, viruses are not affected by antibiotics. Antibiotics only work on living things. They're anti-life. They work on bacterial infections. But this can be a target because we don't have reverse transcriptase of drugs to try to shut down the virus. And many of the successful HIV cocktails incorporate a retrovirus inhibitor, a reverse transcriptase inhibitor, to try to shut down the virus reproducing. Right? Now, virology is a huge field, and there's so many different kinds of viruses. So if this is something that's interesting to you, you can read more about that. You can get a whole degree, a whole PhD in virology. And these two tables, these two slides, just show you some of the different kinds of viruses and some of the variety that you see in their shapes and sizes, whether they have an envelope or not, their genome structure. Is it a single strand of DNA? Is it a double strand of DNA? Is it a single strand of RNA? Is it multiple strands of RNA? Is it single-stranded RNA? Is it double-stranded RNA? So you can see that there's lots to study here. And then if you want more information, do dig deeper and learn deeply.